So my name is Anvesha Mohanty, and I'm going to talk about an intelligent diagnostic system for classifying dermatological condition using computer vision. So in this talk, I am going to cover a brief introduction about my research, followed by the challenges in skin disease treatment. And then I'm going to talk about a specific skin condition which I'm going to look at in this research is called rosacea. Then I'll be talking about key challenges in medical image analysis, followed by aim and objective of this research. And there are a few challenges of rosacea dataset in deep learning models. And after that, I'll be talking more about my literature review on the 5 topics of which are data augmentation and transfer learning, GANs, meta-learning and future classification, 3D face modeling, and explainable AI, followed by work that I have done so far, and then research hypothesis questions and my research plan and the next steps in future. So skin diseases affect 1/3 of the world population by making it 4th most common cause of non-fatal diseases worldwide. Skin diseases cause discomfort in day-to-day life, get worse with time, affect productivity, can be deadly if not treated on time. So a fast, accurate, and low-cost system for diagnosis of skin conditions is desirable. And accurate detection is vital for early treatment and medication as well. However, data scarcity is a central problem when it comes to the skin disease analysis or any other medical condition analysis using deep learning, computer vision, or AI models. So the main objective of this research is to explore the possible techniques to improve diagnosis with a small data set. So here are some techniques to deal with the data scarcity problem that I mentioned. So I'll be looking at GANs to generate synthetic data set, meta-learning and future learning to optimize the model to learn with fewer examples, then 3D face modeling using Gaussian process mappable models to model synthetic 3D faces with rosacea, and finally, looking at few literature on explainable AI for making the diagnosis understandable. So as you can see in this slide, there are 2 kinds of skin disease images, such as dermoscopic images on your left side, which are collected using dermatoscope, which is a medical device used to commonly examine the individual lesions on the skin. So mostly dermatoscope is used for the cancerous lesion data collection. Whereas there is a clinical images on your right side of the slide, which are taken using mobile phones and cameras. So in this research, we are going to look at the clinical images. So here are a few challenges in traditional treatment of skin diseases by human expert. For example, skin diseases are it is very complicated to diagnose because of the observational analytical complications. And this requires years of experience and expertise, making it time consuming and costly. Skin disease diagnosis is a basic need in developing countries. Even in the developed countries, there are only few expert dermatologists are there. But there are few skin conditions which are chronic skin condition that we call, requires a lifelong engagement with a dermatologist by routine checkups and regular treatments. So one of the chronic skin conditions that I'm going to look at in this research is rosacea. So rosacea is a chronic facial skin condition that goes through the cycle of fading and relapse. It is also a skin condition that is related to vascular disorder, a common skin condition in people with fair skin and Celtic origins as well. So according to a report published by National Rosacea Society, 415 million of people are affected by rosacea worldwide. So here are 4 kinds of rosacea when it comes to the medical diagnosis approach. Rosacea is classified into 4 subtypes, such as the facial flushing and persistent redness on the cheeks of a face. So that is subtype 1. And in subtype 2, there are some bumps and pimples, raised red patches on your cheeks. And then the 3rd subtype is a phimosis, which is generally an enlargement of the nose. And the 4th one is ocular kind of rosacea, which is generally eye irritation around the eyes. So there are a few challenges in medical image analysis when it comes to deep learning models or AI techniques. such as there are lack of sufficient images in the medical image analysis problems. And so which generally leads to a long tail distribution of the data set. And there are fine grained variability issue due to similarity in skin condition, and then which may lead to the poor generalization for novel or rare classes. For example, one example is rosacea as well. So as we generally expect medical image diagnosis system to function very well and give us good results, that needs a lot of rigorous training and repetitive training as well, which is really a time-consuming process. And the most important part is these are lack of transparency. So we don't know how this model reached to its diagnosis or the output. So the aim of this research is to explore methodologies for dealing with low data by generating synthetic data, adapting models trained on other domains to the low data domain, and make black box model explainable. So, but here are some challenges of rosacea dataset in deep learning models. For example, we have only 182 images acquired from internet. Here are some examples given as images. And there are 70 images, which is referred as UCD dataset, provided by Professor Frank Powell from University College Dublin. And the main motivation to deal with the limited dataset in this research is the limited availability of dataset for rosacea. So here are the 4 objectives of this research, such as the objective 1 is generating synthetic facial images with rosacea using GANs to increase the volume of the dataset to support rosacea diagnosis using AI, applying meta-learning and few-shot learning to deal with the available limited dataset of rosacea and similar skin conditions. Number 3 is creating a 3D model of human face using Gaussian process morphable models with possible deformations due to rosacea with existing 2D images. And the final one is navigating through the decision-making strategy of deep neural networks in order to make clinical diagnosis interpretable using explainable AI. So here I'm going to talk about few literature review that I have carried out so far to carry out further research in this PhD topic. For example, data augmentation and transfer learning. Data augmentation is a technique to artificially create new training data from existing training data by slightly modifying them. Transfer learning is a process of improvement of learning for a new task through the transfer of knowledge from a related task that has already been learned. So here are some major takeaways from the literature review that I have carried out on data augmentation and transfer learning, such as most of the work done on data augmentation and transfer learning starts from 2017 when it comes to skin disease analysis specifically. And the majority of skin conditions that are considered for these techniques, which are subtypes of skin cancer, such as malignant melanoma and benign nevi. And all these images are the dermoscopic images, as these are the cancerous lesions. But in this research, we are going to look at only the clinical images that I have shown you as an example in the previous slide. So only a few studies related to rosacea have been conducted so far that I have found from the literature review. And most of the work has used the data volume of at least 1,000 images. So here are some research gaps which might be helpful in this research. And further, that CNN models trained on the non-medical image dataset, which are ImageNet, have been used for medical image analysis, stated by Morid et al. And Morid et al. And Pan et al. In their work, they have shown that they have stated that negative transfer is an open problem in transfer learning. And negative transfer problem is supported by the fact that the well-performing CNN models are pre-trained on non-medical ImageNet dataset. So let's look at what are GANs. So generative models are inspired by the unsupervised learning approach. So generative models can generate new examples that are similar to the training images. The GAN framework consists of a pair of adversarial networks such as generator network and then discriminator network. The generator network tries to transform random noise from the prior distribution over the input variable, which usually comes from the standard Gaussian distribution, to generate synthetic images which look as realistic as possible. And then the discriminator network, which simultaneously target to classify between the sample images obtained from the real training data, and the synthetic images obtained from the generator. So by utilizing the feedback from the discriminator, the parameters of the generator can be adjusted such that the samples are more likely to fool the discriminator network while performing a classification task. So that is the idea of GANs. So based on that idea, I have looked at a few literatures on skin disease analysis using GANs. So most of the work on GANs for skin disease classification is from 2018 and after. And most of the diseases considered for this generating synthetic image dataset are melanoma and cancerous skin lesions again. So, so far no work related to rosacea or any facial skin condition have been carried out using GANs. Then minimum of 2000 input real images are considered for generating synthetic images. So here are a few limitations that will be useful in this research and further as well, such as DC GAN and Lab GAN work very well for generating synthetic images, while they are very low in resolution, for example, 64 by 64 pixels. So a conditional GAN can actually help to improve this situation. However, Bauer et al. In his work says that images prone to the checkerboard artifacts, so it can be easily identified as fake. And Lab GAN images are, in the Lab GAN, you can see the images are as realistic and diverse, while the close inspections shows a vast range of high frequency artifacts present in these images. Well, there are a few advantages using GANs on skin disease analysis that I have got so far on progressive growing of GANs, in short PGAN. So PGAN generally gives outstanding results for realistic image synthesis of faces. However, the resolution is 1024 and by 1024 pixels, which is a very good resolution, which can be actually incorporated in this research. And there is no need of conditioning. So it can be trained by ignoring the presence of different classes as well, and the generated samples generally seem highly realistic. So PGAN will be a very good technique to incorporate in this research. So a good machine learning model should be trained over a large number of different learning tasks, simultaneously optimized for the best performance over this distribution of tasks. So a good machine learning generally works well when provided with a huge amount of training data. In contrast, humans learn with only a few examples. Meta-learning is a concept of how to learn, learning how to learn with a small amount of data. So meta-learning model is trained over a variety of learning tasks and optimized for the best performance on a distribution task, including the potentially unseen tasks as well. So each task is considered as one data sample in meta-learning model. Meta-learning refers to learning algorithms that learn from other learning algorithms. So basically it is an optimizing process. So few-shot classification is a subset of meta-learning, and it aims to train a model using only a few data points and training iterations. Few-shot means learning a large number of tasks using dataset containing only a few examples known as shots. So training 5 different classes where each class has just one example is called one-shot 5-way classification task. For example, in this image, it is actually a meta-learning setup where each gray rectangle generally separate tasks that consist of a training set and a test set, separated by the dotted lines, as you can see, the training and test. So each image is taken as one example within the tasks, and each training set has 5 different example images, and each test has 2 different example images. This means you can train the model with a wide range of example of tasks and then test it on the completely new task in the meta test set according to your preference. So the meta test set is also known as a support set. So there are some major takeaways from existing studies on meta-learning and future classification, such as very few studies have applied meta-learning and future classification to skin disease and medical image diagnosis systems. Some studies have shown promising results, such as by using less than 500 images and about 100 images per meta-testing class. And then there is a very good example by Lee et al., which they have got 83.3% for accuracy for 5 samples per class, as I have shown in the previous figure. So there is a work by Wang et al. Where he states that real-world computer vision tasks with low data situation are the first test bed for future learning algorithms. So let's look at how we can do a 3D face modeling using Gaussian process morphable model. Before getting into Gaussian process morphable models, let's look at SSMs, which are statistical shape models. The better the model represents the structure of the objects to be analyzed, the easier it becomes to fit the model. And for this reason, statistical models become very popular while modeling faces and other biomedical images. So as you can see in the left side of the figure, the eigenvectors of the covariance matrix represent the direction of the principal axis of the ellipsoid here, and the eigenvalues represent the squares of the standard deviation along those axes. So and in the right side of the figure, as you can see, the eigenvalues generally drop towards 0. We can restrict our attention to those limited number of eigenvalues which are significantly greater than 0. So that is the idea of SSM that takes further. For example, in PCA-based statistical shape models, the generate sample faces using Gaussian kernel from 200 training example faces, for example, these faces. And we may not go beyond this point in exploring the slide deformations that we can do in the sample faces. However, the generalization of SSMs are called Gaussian process morphable models. Because because the statistical shape models comes with the drawbacks, such as shape variations in statistical shape model is restricted to linear span of training data. Hence, a lot of training data is needed, which is we don't have really. So Gaussian process morphable model can actually help in this scenario. So the Gaussian process morphable models generally start with the standard reference shapes, such as like this. And the Gaussian process morphable models generate a random Ward function and apply to the reference shapes, as shown in this figure. The Ward functions are generated from Gaussian process with a mean function and a covariance function, and you are free to choose which covariance function could be used. For example, in this figure, so here are a few examples of Gaussian process morphable model with manually constructed deformations, such as in the above 3 sample faces with a higher scale factor and a standard deviation, which lead to these sample faces for global deformations, as you can see in these 3 faces. And by choosing a smaller value scale of scale factor and the standard deviation may lead to the local deformations, like it is shown in these 3 sample faces. So this idea of generating local deformation can actually help modeling the slide deformations found by the skin diseases on the face. For example, in these pictures, as you can see, these erupted lesions, the enlargement of nose in another 3 pictures. So GPMs can actually give us the modeling power to model all these fine deformations only when they are needed. So GPMM is generative, so which we can assess the validity of our prior assumptions by sampling from the model. So it may help in incorporating the medical expert knowledge in order to model slide and local deformations, such as lesions, erupted lesions, and the enlargement of nose on the face. So as AI becomes more advanced, humans are challenged to comprehend and retrace how the algorithm come to a result. The whole calculation process generally turned into, which is commonly referred as black box, that is impossible to interpret. So So basically the idea is, how do we know how an AI algorithm arrived at a specific result? So explainable AI is a methodology which can help in large scale implementation of the AI methods with fairness, model explainability, and trustworthiness. So here are a few literature review that I have carried out, and I will be exploring more literatures on this point, such as a deep RAID algorithm, which can extract rule from deep neural networks when an arbitrary number of with the arbitrary number of output neurons. And there are three kinds of taxonomy of explanation methods in deep neural networks, which are rule-based extraction methods, attribution methods, and intrinsic methods. So here is an example of how explainable AI can help with investigating the decision-making strategies in skin disease diagnosis by providing interpretable outputs, such as we can understand why and why not we got some specific output, and why this models get the successful results, and how it fails, why we should trust this model. So based on the literature review that I have talked about on data augmentation and transfer learning, so that those were the first test bed for the few experiments that I've carried out. So I have carried out some experiments using baseline sequential CNN, then InceptionNet V3 with UCD dataset, and a few images from Celibae dataset. And all these models are actually overfitting. And therefore, I decided to explore more machine learning and computer vision methods, which may help dealing with a small data set. So I have been experimenting with the GANs as well, but due to unavailability of the machine and proper computing equipments, I have been just waiting. And now that I have the equipment, so I can proceed with the experiments after this. So the research hypothesis for this topic is for a digital diagnosis system for rosacea and related skin conditions based on the computer vision and machine learning that can provide an accurate and explainable diagnosis for clinicians and patients. So here are a few research questions that I'm going to look at, such as, can GANs provide an adequate solution to the scarcity of data on rosacea by generating realistic synthetic images of rosacea lesions? Can a CNN trained with the synthetic images classify different skin conditions accurately? Can meta-learning and future learning algorithms help train a CNN model to classify different skin conditions accurately with low data scenario? How accurately a 3D model of the human face with facial skin conditions, which is rosacea, can be constructed from a set of 2D images? And what are the strategies to find the decision-making strategy in deep learning models that can help making the model explainable? So here is my experiment plan 1, in which I'll be generating some synthetic faces with rosacea using GANs that can be implemented in various ways, such as in experiment 1.1, I'll be generating new synthetic images from the dataset of rosacea using PGAN and StyleGAN due to its advantages that I have discussed about the PGAN. And then in experiment 1.2, in which I will be generating some synthetic images that can be generated by merging two datasets, such as the dataset of normal face, and which comes from Celibae dataset, and the dataset of rosacea faces, and then applying PGAN and StyleGAN architecture on that to see how I'm getting the results, the synthetic images. So this task may offer a comparative study to assess the precision of synthetic samples compared to the real samples. And in experiment plan 2, which is based on the research question 2 and 3, so this is based on the designing a model using the original dataset of rosacea and a meta-learning approach. In this task, a few publicly available datasets such as ST198, 128, DUM7PT, Edinburgh dataset, LearnNet-NZ will be considered for the training. And then the implementation of a few meta-learning techniques, such as gradient-based meta-learning, such as erectile and prototypical network with G-convolution and two-way classification will be incorporated. So all these methods actually turned to show very good result actually before. So we are incorporating this models in my research as well in future. And the experiment plan 3, which is based on the research question 4, is about designing a 3D model using Gaussian process mockable models, which will be considered in this research due to its various advantages that I have talked about so far. And then there are some various types of GANs based on the concept of statistical shape models and Gaussian process mockable models, such as MeshGAN and GANFIT will also be experimented in this part of this research to see how it construct the 3D faces using a GAN model as well. So it can be compared as well. So here are some project plan and the next steps for the rest of the years of a PhD. So here I'm actually writing a literature review draft, which will be completed by middle of this month. And I'll be submitting those to my supervisors. And there are, based on the few experiments that I have stated, here are a few like milestones for those experiments. And some planned conference submissions and publication plans are there as well here. So yeah, that's it. And here are some references. And thank you very much for listening to my talk. I would like to thank you, my supervisors, Alistair, for helping me throughout this journey till now. And thanks, Maria, as well, for giving me some good feedbacks after my transfer report was done, which was actually helpful for my presentation. And I would like to thank Hossein as well for his valuable feedbacks and